So welcome everyone. It's a real pleasure to introduce Jim DiCarlo, who's going to be giving a lecture today that sort of takes us in a very complementary way to what we've been doing so far. So we've been focusing on deep learning applied to understanding all kinds of things, including, the brain, including many different diseases. What Jim is going to be telling us about is how we should be understanding deep learning with all of the knowledge that we have accumulated on neuroscience. So Jim, take it away.
Okay, thanks, Manolis. Folks, I hope you can hear me. This is really for you. So I have a bunch of slides here, but I'm just going to try to give you a big picture perspective. I'd phrase it a little bit differently here. I think I'm going to phrase this as sort of what does deep learning have to do with the brain, and what are the connections there, at least in the area of visual science. And I'll show you how that's led to progress in both directions. That is, the brain science has influenced much of what's done in areas of deep learning, but it's also benefiting from that. And I'll try to show you that interplay, and maybe if we have time, paint a picture of how that's going forward. So I think of models coming out of deep learning as scientific hypotheses for how aspects of brain function may work. And what neuroscientists and cognitive scientists do is evaluate the quality of those hypotheses with respect to the data. That's how I prefer to sort of look at the interplay between computational science and empirical sciences like brain and cognitive sciences. So the title of my talk, Quest to Understand Neural Mechanisms of Intelligence, another way I like to put this is just to call it reverse engineering human visual intelligence. So I'm going to tell you a bit of what I mean by this idea of reverse engineering, and I hope you'll see how it connects to your course on deep learning broadly. So the goal, and this relates to what I just said a minute ago, the neuroscientific goal of this reverse engineering approach is to ideally account for all aspects of human intelligence at some day. Here I'm just limiting to say human visual intelligence, and I'll narrow that even further in a minute. When I say something like human visual intelligence, what I mean is the behavioral capabilities that we observe and we label as a set of things that correspond to a domain of intelligence, in this case, driven by photons striking your eye and outputs that you make given those patterns of inputs. So that's broadly a domain of vision or vision sensation. And I want that explanation, or many of us want that explanation as scientists, to be in the language of mechanisms. We'd like that to be in the language of mechanisms that we know are physically inside the system that's carrying it out, in this case, the brain, which of course we know is a network of simulated neurons, a network of neurons, and we want to be able, so therefore we want to be able to explain that in networks of simulated neurons. So you're starting to see the connections between deep networks or deep recurrent networks and neuroscience, because we have an example of a deep recurrent network running in each of our heads right now. We just don't know which one it is. And so we want these accounts of visual intelligence to be in the language of engineering, which again doesn't just mean I say there's a bag of neurons in your head, but I tell you about the parameters of that bag of neurons that allows me to have predictive built systems that can be compared against the data and allow us to do interesting things both in brain sciences and more broadly in AI as well. So it's really those three aspects, I think none of which are really controversial. That the one on the left is usually what empirical scientists like brain and cognitive scientists, what they usually start with, which is the observations, whether they're from the behavior, which is broadly called cognitive science, or used to be called psychology, or whether they're from the brain, which is called neuroscience and measurements within the brain. And on the right side are sort of synthesis tools, which come from engineering, whether you're building a deep network, whether you're building a robotic system, whether you're building some kind of algorithm. And what's exciting right now is that these two different approaches, which again are led out of science and engineering, kind of come together around especially deep artificial networks. And these specific artificial networks, you should view as hypotheses of how aspects of the brain function may or may not work. And it turns out that some deep networks are the current leading hypotheses of visual systems, which is what I'll tell you today. There are actually some of the leading hypotheses about auditory system as well, but I won't tell you about that today. So the history of what I'm going to try to give you today is a bit of a history, of background on kind of how we got to where we are, and then maybe point to where we might be headed in the future. Again, I hope everybody's hearing me. Slides are advancing. If anybody has any questions, please just put in the chat, or also if someone could chime in there. Could somebody just give a thumbs up so I know I'm not talking at a wall?
No, this makes total sense, and this sounds super exciting.
Okay, so let's do the history. So first of all, I should say at the beginning, these are folks in my group. Everything I say science-wise, they did. I'm just the ambassador of the work. I may mention some of their names along the way, but any of the research that you see from my lab, it's really their work that I just get to highlight here. So broadly, I already mentioned this, human visual intelligence. So here's an example. You have an array of pixels striking your eyes right now. They're million pixels or so there, striking your eyes. They're just luminance intensities emanating from your screen right now. And somehow you make inferences about what's out there in the world. You make inferences about what's in there. For instance, are there cars? Are there people? Of course, you're all saying, of course there are. And that's a very hard problem, by the way, how you did that. And that's what we've been focused on. But you're probably also able to do things like, what will happen next? And is it safe to walk to the right or should I stay to the left? And other kind of prediction-driven or physics-driven problems that we broadly would call visual intelligence. And I'm putting these are all kind of questions of visual intelligence. It's not a kind of cleanly defined area, but I'm sort of ordering them in sort of maybe the degrees of difficulty as we think about them. And also to tell you that the progress that's been made in brain science is mostly kind of on the what's out there aspect of this. We don't yet have great models of all the things to the right, like predicting where things will happen next and how the brain carries that out. But we're starting to have pretty decent models of how you just estimate what's out there in the scene, like are there cars there, are there people there? And just to kind of narrow things a bit, so when you look at that scene, you may have thought that you took it all in at once, but if you don't know this from biology, you should know that you don't actually have high acuity across your whole visual field. You have high acuity at your center of gaze. So if you just looked at this little red dot right here, and if you just looked at your screen right now, the area of the brain that I'm going to talk about, the set of areas of the brain that cover the central 10 degrees, which is about 2 of your hands held at arm's length. So if you kind of do that for yourself, I'm trying to illustrate about 10 degrees for you here. So we're not trying to analyze how the brain takes in the whole scene, but just that central 10 degrees, which I roughly illustrated here. And the way that you were probably inspecting that scene while I was showing it and talking was that you're making these rapid eye movements that you don't even know that you're making, called saccades, around the scene. You dwell at each location for just a couple hundred milliseconds and then move on to other locations, sampling the scene as you go. So this is kind of how your sensor system your visual system actually takes in these kind of data. And I'm telling you this because it allows us to operationalize the problem from all of scene understanding of visual intelligence to a more narrow problem, which is more tractable to study in the lab and turned out to be a really good starting point. We call that problem kind of what you do in each 200 millisecond glimpse, each 10-degree snapshot that I'm sort of illustrating here now as cutout snapshots. And we've just been focusing on even estimating what's in those snapshots. Is there a car? Is there a person? Is this a sign? I've just labeled this sort of narrower domain core object recognition because we call it core because we think it's fundamental to scene understanding. But again, it's not the full visual intelligence problem. Is a car here? Is a person here? Other questions about the latent content, not just is there a car, but what's the pose angle of the car or the person or the sun? Those are the kinds of questions that we and others have been asking. Again, it's a narrower space of behavior. This allows us to begin to operationalize kind of how you do on those kind of tasks. So here you go. This is an example of you I'm going to test you a little bit yourself. You can just do it at home. If you look at this dot, I'm going to flash up an image for you. That was only about 100 milliseconds, so even shorter than your natural saccadic your fixation dwell time. And I hope you can notice it was more likely a bird than an elephant. So you can imagine I can test you all day long. I'm not pre-queuing you as to what kind of objects I'm going to say. Okay, there was another one, face not a car, and here you go, here's a natural photograph. Hopefully you noticed that was a bird and not an elephant. Okay, I can test you and other primates, other Homo sapiens, like all of us on this call. We test many of them. We measure the detailed patterns of what kinds of successes and failures you have. And one of the things we're really interested in is comparing you to current computer vision systems, again, because some of them are now the leading hypotheses of how your visual system may work in certain ways. And so I'll just think of these as 2 different species here. And we know that we're still better than computer vision systems in a lot of ways. Over the last 10 years, it's gotten a lot harder to see these differences, which suggests that the advances in computer vision, indeed, some of those advances have been driven by the kind of understanding of how our system solves these problems. So that shouldn't be surprising to you that this gap is closing, but not yet closed. I want to introduce to you another species here. This is a rhesus monkey, a non-human primate. It's doing the task that you just did, but by doing a touch screen. So it's triggering the presentation of an image and then choosing among 2 choice objects. Again, there's something like 30 some objects that this animal is working with. Doesn't know ahead of time what object it's going to get. It's just saying, I see something, what did I see? Green means it gets it right. Black means it gets it wrong. You see he's mostly getting it right, but he doesn't always get everything right. And he has both successes and failures, just like you and I do. And we can measure that. There it goes, it got one wrong. And we can measure that all day long, get lots of data. And here's an example of data, behavioral data from us on the left, Homo sapiens, and our rhesus monkey cousins on the right. I don't want to take you through the details of this slide other than to say these are pairwise comparisons of many objects you see listed on the left. The color intensity indicates the difficulty of discriminating those objects from each other across many images. So this is sort of object grain measurements. We have much finer grain measurements than this, but I'm just showing you to give you the big picture message that we can't really distinguish between our abilities and the abilities of our primate cousins there to do these kinds of tasks.
Now, Jim, is this normalized, or in an absolute scale, we're really not much better?
These are absolute scale numbers. And if you look closely, the D primes are slightly lower in primates. They're a few percentage points below us. But I'm here stressing the patterns across the different objects. It's sort of not surprising when you look at this, like knives and wrenches are harder to distinguish than, I don't know, spiders and...
Now, the other question that I have is, how does deep learning fall within the same heat map?
Yes, and I have a slide, I don't have it in the deck, but the current deep learning systems, if I pull up this plot, they are also very similar now on this plot, which is back to the point I raised earlier. But if I look at the image grain, I can see differences between all deep learning systems that we tested and these two systems. So we and our cousins, this is a higher view level here, but as you zoom in image by image, then you still see differences between the systems. And I could pull that plot up if you're interested. But that's the short answer, and I'll set that up.
Perfect, thank you.
Yeah, okay. I was just trying to set up for this group that we have basically a quantitatively nearly perfect model in the rhesus monkey of what you and I do in this domain. And that shouldn't be surprising to you, following evolution. We don't have to rebuild our whole visual systems. We do other things, of course, that monkeys don't do, but here we're studying vision. And it turns out that then we can take this quantitative model. And so here's a big picture you should have in your head that we're basically like a monkey, and we're somewhat better than computer vision systems. Although, as was just discussed, it's harder and harder to see those differences. And that difference right there is extremely interesting to us. So for now, I want you to take this equality here, and it's going to allow us to sort of say, well, what are the brain mechanisms of the human? We'd love to know those, but those are harder to study directly, but we can have more access to the non-human primate. This is the non-human primate brain shown here. And so for most of the rest of the talk, I'm going to tell you mostly about the non-human primate brain, but you should keep in mind that any inferences we draw are very likely applying to the human brain. And that's been tested in various ways. I just won't have time to take you through any more human data. So I'm switching species here to the rhesus monkey, and I hope I motivated that well. And now I want to talk about the set of visual areas that are involved in the rhesus monkey on doing that sort of core visual recognition task. They're not just one brain area, they're a set of brain areas. Of course, there's the eyes that I'm not showing you. There's the LGN in the middle of the brain that I'm not showing you. Then there's a series of cortical areas here that are referred to as the ventral visual stream. So these are areas on the outside, sort of the bottom edges of the brain, shown here, and each indicated in a different color. These have both feed forward and feedback connections across them. Notice they're in sort of a deep stack of layers. It's illustrated here. This is from known anatomy that's decades old. We know things, for instance, that if you lesion these parts of the brain, you get deficits in these kind of tasks. And I want to highlight for you this highest level area called IT, or infratemporal cortex. So you hear me say IT, just think of that as the highest level area across this stack of brain areas, which of course doesn't end here, but projects to other parts of the brain involved in action, decision-making, and so forth. So these are kind of the sort of nearly purely visual areas of the brain. Okay, so I've schematized that for you as sort of millions of neurons in each area down at the bottom, and I'll keep referring to this as we go through the talk. I hope you can see there's already sort of a deep network here, and we should be modeling with deep networks, and then we'll tell you how we've made progress in that regard. But that's, again, anatomy motivated. Okay, what do we know about the function of this area? If I put up an image like this image here, it's of course a pattern of pixels. It's captured in the back of your eye, in this pattern of spiking activity in the back of your eye, and the pattern of retinal ganglion cells. You have about 1 million of them arrayed across the back of your retina that then transmits down your optic nerve through the LGN and the thalamus, and then into new patterns of neural activity successively along these cortical areas, V1, V2, V4, and IT. And I'm just showing this schematically here with these colored circles being like activated versus non-activated neurons. That's an oversimplification in a couple of ways. The neurons have graded activation, but just to fix ideas. And it's also an oversimplification that this is not a strictly feed-forward chain, as I've outlined here, but it's a good first approximation, and it got us a lot of progress from starting from that feed-forward model. So, and I also want to give you a sense of time scale. It's about 100 milliseconds to go from the image striking the eyes to these initial patterns of activity in this high-level area of IT cortex, which, as you might recall, you dwell for about 200 milliseconds. So this is in the right time frame to do things like allow you to pause your search or to make a selection on the next move in your search as you're sampling the scene. And so again, these timescales are matched to your overt behavioral sampling scales as you sample the visual image. If I switch to the new image, a new pattern of neural activity emerges out of IT, again with a lag of about 100 milliseconds. If I presented an old image, then the pattern of activity in IT goes back to the old pattern, again, with a lag of about 100 milliseconds. And, so this is reproducible modulo some stochasticity that I'll show you in a moment. But you can then actually play this game all day long, where you can show lots of images very rapidly, measure lots of patterns out of IT cortex, each image producing a new pattern. And these patterns turn out to be quite powerful, and as I'll show you in a minute. Okay, so how do we know all this? Well, we know this from studying rhesus monkeys and other species while they're doing things like the task I showed you, or fixating a screen such as this one while we show these kinds of images. And then we use recording electrodes, electrophysiology, which are invasive electrodes sampling electrical activity near individual neurons, like this image shown here, where we sample the individual spike waveforms from individual neurons, and we do this kind of random sampling within an area like IT. And let me show you kind of what those data look like, just so you have a feel for the elemental data. If you go and record from individual neurons in IT cortex, remember there's millions of them. Here's just one recording site in IT. Each tick mark in blue here is an individual spike out of a neuron. And you see that this is an image, and here's the spike pattern, and here's the duration of the image, 100 milliseconds that it's on the screen. And you might notice, well, there's a lot of spikes right here at the end. There were a couple here going on here. And if I repeat this image over and over again, you see that, well, there's a little bit of background here, but there's this clear rise in mean rate activity that happens again in a lag of about 100 milliseconds. Notice that it's not identical. So I mentioned stochasticity, but you see this sort of average rise in rate, and I can contextualize that for you by showing you other images. Here's the same neural site in response to 10 presentations of this image. You see, again, a high rate of activity. Here's another image that's sort of a lower rate, but still a little bit of response. And here's another sort of lower, not quite zero response. This is just this is one site and 4 images. Go ahead. I'll pause.
I'm a little curious about the fact that there's less dots on the more natural image of a monkey, and I'm just curious whether they're sort of closer to primitives, so that you need less activation, that their abstraction concept requires fewer neurons to encode it because it's just so natural to them.
These are the kind of inferences that you're making from this data, the kind of inferences that neuroscientists have made for decades. Maybe the monkey's faces are special. Maybe it's all the things you just said. I just want you to, and I think those are inferences that could be made. I think I'd just like you to take these as like, images come in, neural spikes come out. What's going on between them? And we can maybe say more about those inferences, but you shouldn't draw too much from this one example neuron out of millions that I showed you. Let me show you 3 more. Here's 3 other examples, and maybe they'll change your inferences already. Here's another example. I don't know, it likes the second and the third and fourth a bit. Here's another recording neuron, especially likes the third and the fourth. I don't know, Manol, if this is changing your inferences so far, but these I mean, it's just samples out of some feature bank that I'm showing you just to give you a feel for the elemental data. Okay.
It really depends on where you put the electrodes as well, and maybe another monkey is encoding images of their peers in other places, basically.
You're now asking a couple of questions. First of all, it's known that there's spatial organization within IT, that neurons that tend to respond more to things like faces tend to be clumped together. Neurons in general are clumped together. So neurons have similar patterns of selectivity, live by neurons with similar patterns. If I just quantify that as a vector pattern out of a bunch of images, you can show that there's a spatial scale of around 1 millimeter or so where you have similar neurons. They're not identical, but they're similar in those regards across IT cortex. So they're not random salt and pepper. Yeah. And then, yes, there are special areas where there's especially areas of faces and bodies and places that have been studied, especially in humans, that have been identified as special clusterings, perhaps, which gets to your earlier question.
Great, thank you.
Okay, so for now, I just want you to view these as responses out of IT cortex. And you can go ahead and sort of start to quantify that. And I'm going to quantify them for you just to fix ideas. Instead of trying to worry about every individual spike, I'm just going to take the mean firing rate over this window with a lag of 100 milliseconds, put one number behind each of these images. So now we basically have converted all these point process data into just a number, one number for each image for each neural site. So here's 4 numbers for this site. It's the mean spike rate. We can relax that, of course, and change the time window, and we do that in more advanced work. But for today's talk, I'm just going to keep it simple for you here. Think about it as one number. And that now maps to these kind of patterns that I'm showing you here. Again, one pattern of response is almost like binary, high or low, that I'm showing. And of course, there's a graded number that I just illustrated for you. So now here we go. I'm going to now take that IT cortex and show you how we actually get more data than just those example neurons. We do things like putting in these chronic recording arrays, which now have hundreds of sites implanted in each location. Remember, this is still only a sample out of millions of neurons, so we're still very undersampled. Turns out you can learn a lot from just hundreds of samples already, which is interesting in its own right. But what's really cool about these chronic recording arrays is that we can then record from the same neural sites for many weeks or even months. So we can measure thousands of images at each site. So we end up with kind of hundreds or thousands of samples out of each animal in testing many, many thousands of images repeated each many, many times. These are implanted during a sterile surgery. This then is closed up. There's the brain tissue itself there. There's the dura mater. There's the skull. This is all eventually closed back up, and the animal has connectors on its head. Plug the animal in every day, record the neural data while they're fixating or doing a task or showing images, and then you get these kind of data out of it here. I'm now showing you this in green, high response, high green, low response, black. This is those mean firing rates I showed you in a minute. We're just giving one number for each neural site. Now there's hundreds to thousands of neural sites typically along this y-axis here. This is in response to one example image. Notice some neurons are high, some are low, some are high again, some are low. And this is, again, just one sample vector out of IT. We don't record one, obviously. We record, as I mentioned, many. Here's 8 images. Notice when you switch the image that the patterns of neural activity are different. And that's what I sort of told you schematically earlier. And I'm showing you some real data. And then we actually record thousands of images. This is a couple thousand images, so I can't show them all. Here they're grouped by the category they belong to, by the way, and you can kind of see some structure in there. But this is an example of the kind of data volumes that we get from areas like IT and other areas in the ventral stream, on which we try to draw inferences about what algorithmic model is actually running under the hood there in the adult animal. So our questions are really about what's running in this adult system as we observe it, not how it learns to get there. Those are interesting questions too, but it's really what is running in the system at this moment that we observe it. Okay, so what do we know? Well, we know a lot, actually. When you make those kind of recordings, one of the things that we saw early on, and there's some references at the top, and I'll just sort of say this without taking you through the data. If you train linear decoders, so you think of like simple linear classifiers living on top of this feature set here. So you think of this as a feature bank. You have a set of linear decoders, say one for each object category. And if you train them up on small samples of data on the orders of tens or maybe hundreds of images of each object, and then you test their ability to generalize to other images of objects, you get behavioral patterns out of that decode. So now we're going from IT to a decode to a sort of behavioral model, if you will. You get behavioral patterns that are essentially indistinguishable from the ones I showed you earlier for both humans and monkeys. And unless you asked about current deep networks, you get this matrix again. So in other words, you can go from here to here in one linear step with some little bit of training data. You can't, by the way, do that from the pixels or actually any of the other areas here. So this sort of says that this IT feature space is like linearly next to the behavioral pattern. Now, of course, the neural mechanisms are more complicated than just a linear decoder, but it sort of says all the computationally interesting questions of this task have kind of been solved by the time you get to this neural activity. At least that's the way I like to think about those kind of results. And so this allows you, if you're a brain-machine interface person, you're really interested in kind of injecting percepts or the matrix or that kind of thing. These kind of models allow you to imagine if you were sticking electrodes in IT and injecting activating neurons or using optogenetics activating, how you would do that to cause certain percepts. They tell you how you might do that. We can't fully test that. We've tested that a little bit, and things seem to be in line with that idea. That's a whole other brain-machine interface story that I'm not going to talk about today. I'm just pointing it out for you that linkage allows you to begin to think about BMI applications, injection to perception. What I want to talk about, so I just mentioned that. What I want to talk about for today, or the rest of today, is what happens between here and here. So, I'll say, by the way, this is why this is a powerful set, because these are good feature spaces. That means they're powerful as vision systems and as AI relevance. Okay, sorry, skip ahead here. What I want to tell you today is like what's going on from here to here. Look, it's a deep network. It's somehow computed from the image through these transformations. And our question when this is now more than 10 years ago, when we were asking these questions, what's going on mechanistically between here and here? So these are some nonlinear transformations of this image, and we were very interested in what are those transformations. And again, there was a breakthrough in the field, and we were a big part of this around 2013. I'm going to take you through how I think about that story, and this is how you think, how I'd like you to think about how deep networks, deep learning connect to vision science, at least in the ventral stream. I think that was the goal of this lecture. So the big picture, and this is really sort of the guts of it here, that you should have is that there's been a lot of work, of course, guidance from neuroscience that sort of says, a lot of things about this system. These are things about what's going on at the low levels of the visual system. You've probably heard about edge filters in visual area V1. If you've done any computer vision, these are lessons from neuroscience that was known. And these were known to be tiled across the visual field. So you apply a filter at every location that's like nearly identical each location. That's in engineering terms, that's called a convolution. And that, of course, deep convolutional networks are now implemented. That, of course, is standard textbooks for neuroscience. It was also known that you do things like approximate a linear dot product, which again is like a filter like this, followed by some output nonlinearity that might look like this, and you have some normalized gain control. Again, these are like micro mechanisms, biological mechanisms that had been worked out and were known to be part of visual system networks from biology, from measurements of the kind that I was showing you, but morely from low-level areas in the visual system.
Can I ask you a question that I've had for a long time, and I just don't know if anyone has the answer, but for these low-level features of edge detection, when we're doing a deep learning, when we're building these convolutional neural networks, we're learning all of these primitives, all of these filters from scratch. But then when you train them over many images, what you see is that they're all learning these edges, etc. I'm curious for evolved systems like primates and other animals, whether the filters at LGN are hard-coded to be edges, or whether they were relearn the concept of an edge. For example, when my babies are sort of staring at the ceiling, I can interpret that as they're figuring out what are their basic filters at LGN, or they're training those filters to recognize edges higher up. So how much of the filters are actually hard-coded all the way down?
That's a great question. Also, that's a question of developmental neurobiology. And actually, Hubel and Wiesel's Nobel Prize in the late 60s was essentially for that kind of work. There's been a lot of follow-up work. And the answer is, like everything in biology, shades of gray. So a lot of these filters require some kind of experience to make in their full form. But depending on the species, you can see elements of these filters even without even restricting visual experience from birth. And of course, that it also varies, again, it varies species by species. So it's a mixture of something built in and some learning on top of that. That's the short story of that. But it's a complicated, still being worked out question.
Thank you so much.
And when you mentioned the deep networks, I'm not going to talk about this today, but when you train deep networks, you don't have to force this into them. This is things biology learned, but deep networks trained, of course, end up with filters. And part of our ongoing work right now is those filters are hard to distinguish from what we observe in visual area V1, even though they were not hand coded into those deep networks. Wow. And then it becomes a question of what kinds of optimizations can we separate that based on what kind of filters we learned in V1. So that's kind of the active edge of work for us on that particular question. So let me just say, this was more of inspiration for the classes, just to remind them, this is stuff, gleaned over decades, that these things that I'm showing you, this was known from science, and this is what you'd find in all the textbooks. And when I present this all, you should look at this and go, well, look, it's a deep convolutional network. And yes, that's what the neuroscience textbook said. It's just that that's not what computer vision was mostly using for the most part to try to achieve computer vision success. They were using parts of it, but maybe not all of these things. That's not to say that science had all the answers. If it did, it would have already kind of conquered the AI's world. It didn't have all the answers, and I'll tell you kind of how things merged up. So again, I've sort of outlined them for you. Feed forward is a good starting point. It's a deep stack of layers, distributed codes that I've been implying throughout here. And that led, as I mentioned, to people building models this time. Fukushima in the 80s, this is the first probably deep convolutional network. The first model there. Tommy Poggio, my colleague in BCS, building these HMAX class of models. Students in my lab messing around with these kinds of models, running them on, these were run on graphics processors. This was the golden days of this about 10 or 15 years ago, when that was sort of all cutting edge. And all I want to say is this is critically important to science. If you're a neuroscientist, you're like, what does this have to do with, these are hypothesis builds to me. So each of these models is somebody trying to build a hypothesis of how it might work that can be compared against the brain. Now, that's noble, but unfortunately it didn't work yet. So when you did this for 30 years, really guided from that kind of neuroscience data, and here's some of those hypotheses, they really didn't work. They fell short as hypotheses about what's going on in the visual system. How do I say that? Well, there are many ways they fell short, but the one that's sort of most important to us is that they were inadequate. Even though they're mechanistic hypotheses, they were inadequate in that they failed to predict the neural data in V4 and IT. And we can quantify all that by looking at individual neurons and trying to find neurons within these models and asking how well they line up and their response patterns. And again, in the interest of time, I won't take you through that other than to show you a kind of typical example. The black here is the response of a neuron in V4, which is this mid-level visual area in response to a whole bunch of images. And you see it bounces up and down in response to these different images. Those are those vectors I showed you earlier in green. And here's the model's best prediction of that. And I hope you can see it's pretty terrible here. And again, that's what's quantified here. We worry about reproducibility and all kinds of things. This is cross-validated, all that kind of good stuff. Without taking you through that, this is a failure. It's not a complete failure, but it's pretty bad. And area IT was even worse. And then, so sort of turning to like, okay, what changed? Well, the way to think about this is that the problem is that the ideas, the architectures from neuroscience were not really wrong, and it turned out to be correct. It's just that most of the parameters of these models were not known to neuroscientists. They couldn't be extracted, say, from a primate brain directly. So they weren't determined by the brain science results. And instead, the kind of collaborative breakthrough came, and these are a couple of folks that were working in my lab, Dan Yeamans, who's now a postdoc, who's now a faculty at Stanford, who was a postdoc at the time. Ma Hung was a grad student at the time. We basically implemented this loop here, which is the main takeaway I want you to take from my lecture. And it was the main paper I gave you today to read, the first paper. I'll just summarize it for you here, is that the idea is you take artificial neural network like this one, which again, this was not new. This was inspired from decades of work. They're not, these are standard linear, nonlinear networks stacked up this way, which are now common deep convolutional neural networks. This was inspired as a macro and meso architecture from brain science, at least guided by that. And then we had this, what we did that was we thought changed the field here is instead of just trying to fit neurons in here, we said, let's get this thing to do something. Let's get it to do core recognition, which was the task we thought it was doing. That's detecting objects across changes and transformations, the problem I set up at the beginning. And then engineering comes in and says, okay, you want this to happen? Well, we need to optimize a bunch of unknown parameters to get this to work. In the original work, we were doing optimization in a particular way that's not as common right now, and I won't take you through that. In the later work, and probably what you learned in the class is most of the time this is deep learning now. So you're optimized from a loss function at the top, you optimize all the way down. Backprop, standard backprop going down with a bunch of details here, optimize all the micro parameters of those filters while you're to get it to do something like this task or whatever task you want it to do. I don't view this blue part as part of biology. I view it as engineering as a tool to achieve something that biology achieved by other mechanisms. So that's the way I prefer to look at this. But it allowed us to fix parameters that we weren't able to measure scientifically that produce each time you do an optimization. In my language as a scientist, it produces a new model, a new hypothesis about what might be going on in the visual system. You stop the optimization. You can take this thing as an adult monkey, if you will, and you can compare it neuron by neuron with the neurons you're measuring in the monkey. So each optimization run is like a new artificial ventral stream. That's how I like to think about it. And the discovery, the breakthrough, if you will, is that when you do this, so now you bring in both science plus this engineering optimization, these internals of these models start to look a whole lot more like the internals that we were measuring in the monkey brain that I said before the earlier models were failing to recapitulate. And so that's the big picture take home you should have from this lecture. And the idea I'll just take you through, just show you a bit of examples of how this works is how do I compare a model to a brain? So here's the brain at the bottom, here's the model at the top. Remember, it's been optimized, all the parameters are fixed, so I'll call it a neurally mechanistic scientific hypothesis. And then we can compare, and this is what's great about this as a model family, is these are not probability model hypotheses. These are actually neural networks. So that means their physical instantiation can be compared at the grain at which we measure the internal data of the system, whether it's V1, V2, V4, IT. We can make those one-to-one comparisons, which allows us to ground the models empirically and then guide forward progress. So that's a very important part about this class of hypotheses, not just that they work well for computer vision tasks, but they are good as neurally mechanistic hypotheses of what might be going on. So how do we make those comparisons? I'll just show you this just to give you a sense of like, here's a response of a site out of IT. Remember, that's this data volume I showed you earlier. This is one neural site across thousands of images. You see the response to each image here. This is not time. These are different images. Here's some example images. They're just grouped by category here. And now here's the model, and then we go to its level of IT, and we pull neurons out of this IT level, and we compare them. And there's several ways to do that. The simplest one to think about is just find the neuron that individually looks most like this neuron using some of your data, and then check the predictions on held out data. And that's kind of what I'm showing you here, are predictions on held out data. And you see that this looks very good in terms of matching the detailed patterns of response of this particular neuron, as if we sort of recapitulated this IT neuron in this model, even though we hadn't fit the model to match the data. It just emerged from this optimization procedure. Notice it's not perfect, but it's far better than the models we had before, again, that I showed you as failures. So this is the breakthrough in our mind that I wanted to tell you about. You get this explanations of V1, or all the levels. I'm showing you IT, but it's true of all the levels. I'm just not showing them all to you here today. So the summary, the big picture is that you have now in silico ventral stream neurons that turn out to be very functionally similar to the biological ventral stream neurons. And I'm going to skip through this in the interest of time. It's basically saying that optimizing for performance led to gains in matching the brain. And so this is now a kind of continuing cycle for us going forward, and maybe a point of discussion as you think about optimizing further models of what can go right and wrong in that. I just want to kind of get towards the end of the talk here, just so we have time to just discuss if we do have any left, is just to say, look, think of these models as hypotheses that I've been illustrating. They come from science and engineering, but done with the influence of science, they can become hypotheses for how the brain actually executes some of these sensory system processes and other processes as well. So I mentioned vision, I already mentioned audition. Josh McDermott's lab here at MIT is someone to really pay attention to there. There's others working in these other areas where they're taking a similar style of approach here if you're interested in other questions in brain sciences. Of course, these models, as you know well because you're in this class, they have influence in engineering, and now this is kind of what people call, quote, AI. I don't like to call this AI, but this is an example of something that is being used as AI at the moment. We're now, instead of just doing deep inference for vision, the ideas of deep learning can be applied as sort of nonlinear optimization on many, many data-driven problems. And that's, of course, what broadly is called AI at the moment. And people are using that hammer all over the place, as you know. I hope I've contextualized how I think of deep learning and deep networks as they relate to vision science. And now, of course, what we're interested in is these are hypotheses, but they're not perfect. So now we want to use them to start to guide further science and to even optimize those models even better, which we think leads to even possibly further engineering payoffs. So the big picture you should have from what I've told you so far is like there's been great progress. The glass is half full. These models as hypotheses of how the brain works. And I tried to give you a sense of how we arrived at them. And when I say how the brain works, I mean on one domain of one aspect of visual intelligence, just visual object recognition, core visual recognition. But none of these models are perfect. And we can see their discrepancies. And those discrepancies are to us the juice to guide new model updates to try to fill up this glass. And we track that. If you're interested in submitting a model or comparing against brain data, anyone in the class who has a model wants to compare, please go to brainscore.org. You can upload your model. You can get scores on how well you match it, kind of the data that I've shown you and how well you're doing as building a model of brain science. And you may have the best model out there, and that would be great. And we'd like to try to acknowledge you if you do that work. And please just go to that site. So I think I've said all this, and I think in the interest of time, I'll sort of end with this slide here and say, look, the forward-going questions is, well, what can you do with these hypotheses? Why do they count as an understanding? And I have things to say about that. And maybe you're probably curious, like, how are we trying to improve these going forward? And of course, I have things to say about that, and that's ongoing work in the lab. But I think since I've noticed I'm sort of near the end of the time, I'm going to just jump ahead to my conclusion slide because I don't want to take away from the next speaker here. So let me give you some take-home messages of what I tried to tell you today. So the background is that the ventral stream produces, that area of cortical brain areas produces a population representation that carries information that's linearly decodable, image generalizable solutions around core recognition tasks. And I showed you those colored matrices where you map the IT data to explain the behavioral. So that was the first thing I told you. Then I explained to you the overall story, and this is what's in that review article, is that if you optimize some deep network architectures for core recognition tasks using engineering optimization, and then you freeze that network, the internal representations of those ANNs turn out to be remarkably similar to the internal representations at all levels of the ventral stream. And I showed you one example of one neuron on that. This includes IT neurons that are specialized for things like faces, like that Emil's brought up in the questioning. I want to stress this point. This result is consistent with, but does not imply the brain learns by backpropagation. It just, think of it as an optimization tool that converges to what evolution has given us in this network that may be optimized in different ways, both through the genetics of evolution and unsupervised methods that Manolis was also actually referring to in your question. These models, and I didn't show you this because I didn't have time, they can start to do things like, and allow us to do things like hyperactivate neurons deep in the brain. So if we have these models, we can actually start to do really cool things within the brain from controlling the pixel level that we couldn't do without the model. So they act as because they're such a good hypothesis, they allow us to do things even in their non-perfect form. These are all things that I think are progress that I'll tell you about. But I'll tell you, sort of, there's lots of things that need to be fixed. As I mentioned, they're not yet identical to ventral stream. Even the best models that we've checked in terms of brain alignment, for instance, they don't have a lot of recurrent circuitry. And we know that that's an important part of the process that I gave at the beginning. I sort of outlined the anatomy, but we know physiologically that's critical too. And we have some recent work on that. And again, all this means we got more work to do as a neuroscientist. We got to get some more data. We got to build some more models. And we think we can converge to more and more accurate models that lead to payoffs in both AI and also just understanding how our brain works as a sensory system, especially a visual intelligence system. With that, I'll end. And if there's any time, I'm happy to entertain questions. Thanks.
Wow. So this was truly phenomenal. I want to ask everybody to join me in really thanking James here. That was truly spectacular. So it's both disappointing and exciting that they match. On one hand, one would wish that somehow we could eventually surpass human intelligence, but maybe the fact that we're even close suggests that it's because we cheated. And we kind of, built on what is truly millions and millions of years of evolution, not just of primate visual cortex, but, eyes and, visual perception, etc. So that's sort of one comment. The second comment is regarding the cognition, where a lot of the same types of architectures as the visual system have been sort of repeated in the frontal cortex and in sort of the auditory cortex and the motor cortex and so on and so forth. So I'm just curious if that general architecture may have evolved in the visual space and now been reused in other spaces, or whether this is, in fact constraining the way we think, and more interesting sort of thinking architectures from machine learning and AI can sort of take us to other modes of thinking, or whether this is truly a well-suited architecture for the types of world that we live in and the types of data and inferences that are sort of well-suited for the world that we inhabit. So I'm just curious if you could sort of tell us a little bit about sort of the, what your current view is, and you don't have to have obviously all the answers because the field doesn't have all the answers, of sort of where do we stand on innovation? Is there room for innovation in deep learning beyond just trying to mimic more of what our brain has already evolved? And can that take us to, not necessarily superhuman, but different than human type of intelligence?
Yeah, there's a lot of great questions in there, Melissa. I'll just try to summarize for them. So first of all, when you say deep learning, I hear, optimization of some architecture. So I think most of the innovations on the brain sciences will come from the choice of architecture, and the optimization tools will hopefully be matched or available to then allow that optimization, which again, I view as filling in for the things that we're not going to be able to measure. Like, I think there's a lot of work to be done in like different architectures with optimization on top of it. The architecture I showed you is only a feed-forward architecture. And when you talk about cognition, of course, you start to involve reasoning, memory storage, things that are going to involve time, right? So I think the way forward for this is to just say, feed-forward networks get you the feed-forward paths of the visual system. But those recurrence questions that I mentioned that we can study in the visual system, especially in monkeys, may give insight to things that are kind of subconscious to us, but are the elements of what you might call cognition down the road. And I think I just want to stress that a lot of our visual processes, again, they're automatic, right? I know we like to focus on the things that feel like our reasoning, but a lot of our power comes from those subconscious processes. Yeah. So one thing I really want to say is that these areas, cortical areas, anatomically, we know the brain looks the same. You go to other areas and you zoom in on the anatomy, it's not like there's a new structure out there, right? So your question about does the things extend forward? Anatomically, the answer is yes, but we're only modeling a kind of portion of that anatomy is I think the way I like to think about it. So I hope you can see how the anatomy connects the ideas of recurrence, and then that might connect to sort of downstream areas that are involved more in cognition as you refer to it.
But I have two more questions that are coming from the students mostly. I'm going to rephrase them slightly. So one of the questions is, how much feed forward is there? So we saw sort of residual networks as one of the architectures that has been very powerful in image recognition, where some of the layers are sort of projecting forward several layers through. And one of the questions was, how much interconnection is there beyond these proximal layers? And how many of these long-range projections are there? And how much of a role do you think, or does the field believe they're playing?
Yes, that is another great question. And I could show you a slide on that that shows you some of the anatomy, but I'll just try to summarize it because I don't see my slide handy. But that is basically everything I showed you was like an oversimplification. There's skip connections across those areas. There's a bunch of brain areas ringing around the ventral stream that are recurrently connected with those areas. And so it's not as simple as just pulling out the piece I showed you, the ventral stream, and studying it in isolation. So that is the work that's going on now. And if I had time for another talk, I would sort of show you how we're trying to approach that. It's basically silence connections, try to block connections, then look at the effects of what happens to the system. But I think I don't know if I deviated off your question, but maybe I've lost the thread, Knowles, but there's a lot of recurring connections, and we are working on that, and it's a hard problem.
Perfect. The last question is, so this question was by Chris Parsons. The last question is by Bo too. So he's basically alluding to the concept of neoteny, where, for example, primates take a lot longer to develop and humans take a lot longer to develop, where our brain is very malleable and we're still learning these functions by being exposed to the world, which perhaps has allowed humans to adapt to, dramatic evolutionary and technological innovation. By contrast, many animals, as soon as they're out, they start walking and running away from lions, etc. And my question, the question that we're asking is, do you see that their proportion of hard-coded functions and filters, even in the visual cortex, is perhaps higher as you go to sort of species that we think of as more basal evolutionarily, and sort of perhaps less hard coding for the sort of bigger frontal cortex, bigger sort of, cortex species. So if you look at, the primate brain, the human brain, and the, sort of lower brains of the vertebrates and, before that even, I'm just curious if you could sort of comment on the proportion of hard coding and how much is known about that.
Well, the way I like to think about that is, of course, the visual areas, like both of our retinas are probably hard-coded. Like across species, our retinas are pretty stable, right? But the further up in the system you go, the more plasticity you see. And monkeys have areas up to some level, and then humans, the extra brain tissue is kind of like, if you look at that visual system in the monkey, it looks like half of the monkey brain or a third of the monkey brain. And the human, that's squeezed way back. It's only like, it looks like a tenth of the human brain, right? So all those downstream areas, if you follow that, there's just going to be more. You don't have to change the plasticity of the areas I showed you to still have a bunch more flexibility, because you have a whole lot of other areas that the monkey doesn't even have, right? But I don't know if the areas are intrinsically more flexible or it's just their positioning within the network. That's another question. But that's how I conceptualize where we are right now, is like there's plenty of room for flexibility that doesn't require. You should be happy to think we all have a primate system running in our heads, and you should be okay with that. Right? You have evolution, but you have other stuff. Part of you is monkey and part of you is beyond, and that's okay, right? I mean, that's how I like to think about it, and that allows us to study it in a way that we couldn't study it in you and I.
Well, thank you for the multi-layer blowing lecture of all of the layers of my mind have been blown here, and I think I want to thank you on behalf of all of the students, because this really reshapes our view of the whole course so far. It's just such a perfect way to have the last lecture. So thank you, Jim, and then I look forward to many more conversations. And,
thank you for having me. Thank you all for attending. All right, I'll sign off. Thank you.
Awesome. And we are very fortunate to also have Jason Kim, who will tell us about the work that he and Danielle Bassett's group are doing in this area between deep learning, machine learning, and neuroscience. So Jason, take it away.
Hello. I am going to share my screen. Can everybody hear me?
Yep.
And everyone is seeing the right screen?
Yep.
Nice. So hi, everyone. Thanks for having me. My name is Jason Kim, and I'm a PhD student in the Complex Systems Lab led by Dr. Danny Bassett at the University of Pennsylvania. And it's going to be basically impossible to follow up that talk by Jim, but I will do my best to sort of carry forward some of the ideas that he built on. And I'm here to talk about some work thinking about brains as recurrent neural networks, as Jim's talked about. And of course, if anyone has any questions, feel free to interrupt and ask. So true to the name, neural networks are just that. They're networks of neurons. And as you have been learning throughout the course of the semester, artificial neural networks come in all sorts of shapes and sizes and are closely related in both form and function to their real biological counterparts. Now, we just heard an extensive, an amazing lecture about this before, but convolutional neural networks and a lot of these types of network architectures were largely inspired by a real biological brain system, such as visual receptive fields. And here in this particular example, single neurons fire more quickly in response to specific orientations or locations of simple visual inputs. But the story doesn't end here, as we had heard. By asking questions at sort of the intersection of artificial and real neural systems, we actually learn a lot about how to train artificial networks and how to understand how real ones work. An example of this kind of question is, how do these receptive fields emerge from training? Where do they come from? As Jim mentioned, how do they emerge from training? And if we take a set of natural images and do some naive decomposition or feature extraction through a method like principal component analysis, what we get are not bases that look like receptive fields, right? Instead, we get these grid-like patterns of increasing spatial frequency. Now, if we want these bases to look more like receptive fields, a simple and elegant solution is to learn not just an efficient representational basis, but also a sparse one. And so if we include a sparseness term in the cost function, the sparse code has a basis that recovers many of the properties of real receptive fields. And these ideas from efficient and sparse coding are still used to study both artificial and real neural systems.
Jason, could I ask you to comment a little bit about the quantification of sparseness? Is it that when you run the features on the same input data, they give different answers? Is that how you quantify sparseness? Or is it sort of from the internal representation of the correlation structure of their weights or something like that?
No, so the sparseness in this case is basically of all these different basis functions. And what you want is when you're representing any given image in the set of naturalistic images, you want to use as few of the bases as possible, right? As opposed to this very spatially regular gradient, you're going to need small amounts of every single basis. This sparseness constraint is sort of to say, okay, well, I want to represent my images using as few bases as possible for any given image.
And how do you compute it computationally?
How do you compute sparseness?
Yes.
So every one of these basis functions or these receptive fields, in order to reconstruct an image, takes a weighted sum of basis functions with some AI coefficient, and we want as many of these coefficients to go to 0 as possible.
Perfect.
And yeah, mathematically, it's just like an L1 norm, right?
Perfect. Thank you. Regularization, basically. That's
yeah, exactly. And these ideas are not new, right? They were certainly figured out a long time ago. But just the point that I want to get at here is that by looking at sort of the intersection of trying to recapitulate a lot of things we see in biological neural networks using artificial ones, we sort of learn principles that generalize to helping us build better artificial systems and understanding what the real ones might be doing. And I think there's a lot of potential at this intersection, and particularly in recurrent neural networks. Now, the power of recurrent networks is that they have memory and hidden state dynamics that offer a lot of computational power. And it's this kind of computational power that we typically look to when we're thinking about emulating the computational power of real recurrent networks. And when I say real recurrent networks, I mean, the human brain, or more specifically, your brain. Yes, you, the one who is listening to this talk right now. At this very moment, your brain is seamlessly integrating visual and auditory information and perhaps multitasking by checking your email or your Facebook or whatever. But more than just parsing the information, your brain can actually change its internal representation of knowledge and memories. So for example, take a few seconds to look at this Rubik's Cube, attend to the geometry and the shape and all of the colors. Now, if I asked you, how do I rotate the cube to solve it? You might tell me to rotate the top face clockwise, but you're not looking at the cube right now as an input. Instead, you're using the cube that you've stored in your working memory and spatially manipulating it to give me the directions to solve the cube. And indeed, if we rotate the top face clockwise, the colors match. So today I want to talk about how we can learn more about artificial and real networks by studying brain-like behaviors in recurrent networks. But the first thing we'll look at is how recurrent neural networks learn to store and modify internal representations and memories. So this isn't some kind of classification task. It is, in some sense, a prediction task, But the goal is once we've learned or once we've taught our neural network how to reproduce an input, once it's sort of stored it in its internal memory, how can we then learn to modify that representation? And then we'll look at how recurrent neural networks are able to separate overlapping signals like we do in the cocktail party effect. And by studying these brain-like behaviors in recurrent networks, our hope is to find principles that, just like in the convolution or feed-forward cases, that expand the capabilities of artificial recurrent networks and help us understand how the real ones work. So let's start with working memory in recurrent networks. Now, as it turns out, real neural systems are actually quite excellent at storing and manipulating representations. And a great example of this is a brain region called the hippocampus. Now in this video, what you'll see is the actual position of a rat moving around along with its decoded position from neurons called place cells, which you've probably heard of before. We see that as the rat moves around in space, the decoded neural activity tracks its position. While this is already pretty cool, what's even more interesting is that the rat can manipulate its representation to plan future trajectories. What you just saw was the decoded neural activity planning a future trajectory before the rat even moved. Let's watch another one. And finally, the rat's actual motion follows the planned trajectory that we just saw. So in addition to these spatial memories, neural networks can also change temporal memories. A great example of this is the songbird. A baby songbird learns to sing by imitating adults. So at 40 days old, this song lacks any particular kind of structure, as you can see in the spectrogram. At 55 days old, we observe some more complex waveforms. And at 80 days old, we see the repeated motifs of a fully mature song. Now, what we can do is we can perform some peculiar perturbations to change the songbird's representation of its song. Specifically, by cooling the temperature of a region called HVC, the songbird actually linearly extends the duration of its song. And I personally think that it's a bit absurd that neural networks can manipulate information like this. In contrast, in a computer, there's this very carefully engineered binary system of representation. So if we wanted to add two numbers, say 7 and 17, we first have to encode the numbers into binary. We then have to modify the encoding in binary space and decode the resulting output. Every step is very delicately engineered, and the failure of even one of these bits leads to large errors. But a neural network has no pre-engineered scheme to encode, modify, and decode information. And so the question I want us to think about is how can neural networks learn this delicate process of manipulating information solely by observing examples? And so to answer this question, we have to make a few modeling decisions. First, we have to create a model for the memory that we want to learn. And like many real memories, we want our model to change with time. We want it to contain some structure, and we want it to be reasonably complex. And a great candidate for such a memory is a chaotic attractor, and specifically we'll be looking at a Lorentz attractor. The Lorentz attractor evolves forward in time according to some ordinary differential equations, shown here, and it generally follows a butterfly shape, but the specific trajectory is unpredictable and sensitive to initial conditions. Now, if we want to store these memories, we next need a model of our neural network. And a common model is a simple, nonlinear dynamical system that is often referred to as a reservoir. So this reservoir evolves according to a simple ordinary differential equation, as shown above, and it's schematically represented below. And you've probably seen any number of neural networks of this particular form. Here, R is the state of the reservoir. In this case, it would be considered the hidden states of the recurrent neural network. X is the three-dimensional inputs that this reservoir is going to see. C is some external control parameter that we'll use later. And D is a random bias term. So in essence, what we're going to do is we're going to sum the contributions of the internal hidden states, the inputs that's provided, the control term and the bias term, and pass them through a nonlinear sigmoidal function G, which here we take to be the hyperbolic tangent. And so, all of the instantiation of the weights are actually random. So B, C, and A are all initialized randomly. And the only training that goes on in this entire process is to change the output weights W.
Can you remind me what the control is?
So the control is how we're going to actually modify the neural network's representation of whatever memory it's learned. So the control can be some signal from some upstream or downstream brain region, but it's essentially effectively going to be a scalar number that we tune and we control up and down ourselves.
Could you give us a practical example of a setting where such a control would change the meaning of something? Can you give us just an example?
So I think there's currently work being done in some neuroscience labs studying sort of beliefs of different task conditions in macaques, and it can be shown that in certain brain regions, the baseline firing rate of a more cognitive upstream brain region tends to sort of fire higher when the monkey expects the task to be of one type, and then firing rate is lower when it expects it to be a different task.
So the control gives you context, and sort of that context can boost, and in the context of expecting something, it can sort of synergize.
Precisely. And, context and boost are exactly the right terms to use. And even in the macaque examples, what they're finding is that it's sort of like, this context dependent firing rate is biasing the network to some alternative levels of baseline activation around which the network evolves. And that's exactly the mechanism that we're going to observe here as well.
But now as you start thinking about learning all of this, basically having this sort of context modulatory ability also means that you need enormously more data for training, or perhaps it means that for training the same types of functions, by modularizing it between the A and the C, you're able to sort of learn a common A, which gets modulated rather than a larger set of parameters. Is that how you think about this as a sort of basically, what are the constraints in the context of complexity and capacity for?
Yes, certainly. So, the adjacency matrix is always going to be n squared parameters. So there's not much we can do about that. When we're thinking about the context variables, we're thinking we're imagining them to be fairly low dimensional things. And so C doesn't actually add practically many more degrees of freedom in terms of like parameter space searches.
Got it. Thank you.
Yep. And most of the examples you see, C is not a vector, but it's a scalar. And so the capital C is going to be an n/1 vector. And so it's going to have minimal impact in terms of the overall numbers of parameters. And all the things you're going to see today are actually just, A, B, and C and D are randomly initialized. And all of the things that I'm going to show you work for these randomly initialized parameters.
Perfect.
Yep. And so the first question we want to ask is, how can we store our memories inside of our neural networks? How can we sort of, get our neural network to evolve about whatever memory we want to see? And previous work has demonstrated a simple, somewhat magical way to store memories in reservoirs. And there are a variety of ways to do this, but the general idea is first you use, the attractor time series in blue, which are your inputs, to drive the reservoir and generate the neural states that we show in gold. And so we take the three-dimensional Lorentz inputs, and we just evolve this forward in time to get R of t, which are the reservoir states in gold. And then all we do is we train linear combinations of the gold reservoir time series to produce an output, the red series, that mimics the input. And we're not trying to learn any lags. This is sort of just an exact replica. Can I use R of t to reproduce X of t using linear combinations W R of t? And here W is just some 3/n linear combination matrix. And finally, to store these memories, we perform stable feedback, and we use the trained outputs in place of the inputs to yield an autonomous system. So it seems kind of like cheating, but you said, okay, well, WR of t looks like X of t. Let's just do some state feedback and use WR of t instead of X to get our prediction reservoir equation that only depends on the hidden states R. And when we plot the trajectory of the true Lorentz against the reservoir output, we find that both systems evolve about a Lorentz attractor. So in some sense, this very simple scheme of just learning to imitate the inputs you've observed is enough to store memories inside of our neural networks. And so now that we've stored the memories, the next question is, how can we modify the memories inside of our neural networks? What does that even mean? And so first, we need to define the modification that we want to learn. And here, what we'll look at is a translation of our attractor in the x1 direction. So in this example, we drive our neural network with 4 translated versions of our attractor. And at each translation, we change the value of this context or control parameter C from 0, 1, 2, and 3. So for these 4 shifted Lorentz versions, every time we show our neural network one of them, we're going to change the value of C from 0 to 1 to 2 to 3. And all that's going to do is it's going to bias the baseline state of the reservoir. And then what we'll do is we'll just train output weights exactly as we did before to take linear combinations of reservoir states or hidden states to reconstruct the translated inputs. And then we perform feedback. And notice we didn't perform feedback on the C parameter. We only did it on the x1, x2, and x3 parameters, which allows us to change C as a free variable. Now before, without the control parameter, what we saw was that the reservoir's memory autonomously evolves about a Lorentz attractor. And once we do the feedback in this case, when we change the value of C, we observe that the reservoir, its internal representation, it has learned to extrapolate and interpolate the translation operation, in this case, just along the x1 direction. And importantly, the only thing we needed to get this kind of behavior was to show our reservoir these 4 examples and tie its experience to the inputs to some context parameter C. And this translation can be successfully learned in any direction, including the x1, x2, and x3 directions. Now, you might say that translating an attractor is not particularly difficult. You can look at an object and sort of move it around in mental space. A much more complex modification would be to actually change the geometry of the attractor manifold itself. So can we change the actual shape of the attractor using this simple scheme? And the answer is yes. So as before, we provide 4 training examples of a Lorentz attractor that's squeezed in the x1 direction, perform training and feedback. And surprisingly, we find that reservoirs can also learn to interpolate and extrapolate transformation operations on its internal representation of attractor manifolds. And these transformations can be fairly arbitrary, whether it be a stretch, a univariate shear, or a multivariate shear. And so at this point, we really wanted to push the limits on what computations these reservoirs could extrapolate. So we thought to give it a seemingly impossible task. Now, in the Lorentz system, as we change the parameter rho, the Lorentz undergoes what's called a subcritical Hopf bifurcation. So for smaller values of rho, the Lorentz trajectories aren't chaotic, but they go towards a fixed point. And as we increase rho to 24, the fixed point becomes less stable. And once rho hits 25, the fixed point loses stability, and we start to get the Lorentz-shaped trajectory that we saw before. So the impossible task was to see how much of this global bifurcation structure can be inferred by the reservoir. And so for the training, we only showed our reservoir examples of stable trajectories that went towards the fixed points at rho equals 23, when we gave the reservoir the context C equals 0, and rho equals 24, when we set the context variable equal to 1. And then we performed training and feedback, and we changed the control parameter to C equals 5. And what we found was that surprisingly, not only did the reservoir correctly predict the local loss of stability of the fixed point, but also the entire global structure of the Lorentz attractor. And we wanted to give it an even more impossible task. And so we showed our reservoir stable examples from only one of the wings, performed training and feedback, and again found that even with this very restricted set of information, the reservoir, the neural network, is able to correctly predict this bifurcation. And there are many more types of predictions that our neural networks can learn. I won't go into too much detail, but basically the Lorentz attractor has several other bifurcation regimes where if rho is equal to 100, then it evolves about a single 1-period cycle. And as you decrease the value of rho, the periodicity of the cycle doubles into 2 cycles and 4 cycles and 8 cycles, so on and so forth, until the trajectory becomes chaotic. And basically, if you only show your recurrent neural network a few examples of one cycle trajectories, then it's actually able to fully reproduce this highly nonlinear period doubling bifurcation scheme. And it can also learn to predict the bifurcation diagrams of several other dynamical normal forms, such as saddle node and supercritical pitchfork bifurcations. And we find that these neural networks can also learn to predict non-dynamical kinematic trajectories. So here's an example of a modified Janssen linkage. And this is a mechanism that sort of has 1 degree of freedom and traces out the trajectory that's shown in the purple curve. And as we change the length of the limbs, say the limb indicated by the dashed purple line, the trajectory changes. And essentially what happens is if you train your neural network on trajectories from a small range of changes in the rod length, the neural network is able to accurately predict and extrapolate the nonlinear trajectories of the much more dramatic changes in rod length. And we also found that if you not just give it one context parameter, but multiple context parameters, then when you train your reservoir on Lorentz attractors that are translated in multiple directions, and you tie an X1 translation to one of the control parameters and an X3 translation to another one of the control parameters, then the neural network is able to sort of independently tune these parameters to sort of move its representation of its memory in space. And so in these examples, you might be wondering what's the mechanism behind the reservoir's ability to learn these computations. I'm probably running a bit short on time, and so I won't go into too much detail, but essentially what happens is if the examples that the reservoir sees are close enough together, then the W matrix, right, that we learned to train the reservoir states to the inputs actually ends up learning the differential relationship between how the vector field of the reservoir changes with the change in the C parameter to the change in the projection of the Lorentz that it wants to see. And you can, go through the derivations and all the whatnots. But essentially, that's the intuition behind it is the W matrix is also learning, it's mapping the differential change in the reservoir response due to changing the context variable to the differential change that it wants to see in its representation. And the same math holds true for transformations as well. As for a mechanism for the bifurcation, what ends up happening is, I won't go into too much detail, but basically recurrent neural networks not only learn sort of continuous changes in spatial coordinates, but also in its own eigenvalue spectra as mapped by the stability. And so basically what happens for the Lorentz system is as you increase rho from 23 to 25, the fixed points lose stability. And what the recurrent neural network does is it actually imitates the specific types of spectral features of top bifurcation along the increase in the C parameter or the rho parameter. And these examples are just that, right? They're examples. In our lab, we decided we wanted a more general way to think about memory formation in these neural systems. And it's not that individual neurons are exactly encoding the independent parts of the input stimulus, right? It's not as if neuron 1 is exactly tracking the X1 variable, neuron 2 is exactly tracking X2. Rather, the neurons form a distributed representation that somehow maps the stimulus to the neural dynamics. And one way to formalize this idea of mapping stimuli to neural dynamics is through the concept of generalized synchronization with respect to some mapping function phi. And the basic idea behind generalized synchronization is that the activity of some neural system, x of t, can be written as some nonlinear map of its inputs, s of t, through a map phi. However, it's not actually enough that we can write the neural representation as a function of the inputs. For a representation to be, truly formed and learned, we also need the map to be invertible. That is, we need to be able to write the inputs as a function of the neural states as well. And the existence of this inverse then allows the network to sort of drive itself with its own representation. And that is sort of the key mechanism behind storing the inputs as memories. And this concept of invertible generalized synchronization is able to make some powerful predictions. So in this paper, a postdoc in our lab, Jixin Liu, was able to get recurrent neural networks to do some pretty cool things. So the theory stipulates that recurrent networks should be able to store many of these chaotic memories, and Jixin showed that indeed this was the case. And so this is one recurrent neural network that is sort of being transitioned, has learned these 4 attractors in different parts of its representation space, and it's able to switch it's able to switch which of the attractors that it wants to evolve along. And of course, with any implementation of a theory, we get some interesting and sometimes crazy behaviors. So when the learning is successful, then we're able to properly learn and stabilize all of the memories. And as you saw before in the previous example, we're able to switch between them with no issue. However, if the memories aren't properly learned, then one of the memories may be unstable. And what this means is that, our neural network might start sort of thinking about whatever it's learned in that memory, but because it's unstable, its mind will sort of wander away and move towards the stable memory. And we even observe some instances of like creativity where sometimes the network's representation isn't perfect, and so it demonstrates some transient excursions off of the expected manifold itself. And so in sum, we've demonstrated that reservoirs can sustain chaotic memories, they can translate and transform memories, and they can infer unseen global nonlinear structure, all by imitating these types of examples. And so there's no learning here outside of just imitating what the neural system saw using the reservoir states itself. And given just this simple imitation, we're able to learn representations and computations, these manipulations of working memory. I don't know how we're doing on time, but
we do have time for a few questions, and I have I have a couple. So this is all fascinating, and one of the things that we looked at in the context of recurrent neural networks in the class is the fact that you can effectively use a time course of data to effectively do self-supervised learning, where you're constantly trying to predict the future. And I'm curious if, and sort of there are many models of how, actual living brains work, whereby you are constantly surprised by, the future, and you're adjusting your internal state of the world, where you're constantly sort of learning to predict and using that as just an immense amount of endless training data, because your entire life is a lived experience of that. And I'm just curious if you're sort of seeing these types of structures, whether you have, what the neuroscience evidence is for these type of feedback learning, and sort of how that applies to the models that you showed us here.
I see. So definitely in terms of neuroscientific evidence for feedback learning, it's a difficult one because this is a pretty extreme learning condition, which is just have this set of output weights that are linearly defined, linear combinations of the neural states to reproduce the inputs. We saw a bit in Jim's talk that at least in certain parts of the visual cortex and the visual stream, we are able to get this linear separability and reconstructability of terms. But I will say that this is just one particular brand of recurrent neural networks. You can reproduce a lot of these similar phenomena using backpropagation through time. And once you do backpropagation through time on LSTMs or whatever, then you run to the question of, okay, well, how neurobiologically motivated and feasible is backpropagation through time in neural systems. So that's sort of the extent to which we've thought about it. Is we can go to more traditional backpropagation through time algorithms, but as far as how biophysically motivated those are, that's a tough question.
It doesn't necessarily need to be backpropagation through time, because what you're doing there is that you're artificially sort of expanding the network, as opposed to sort of having a fixed set of parameters where you're constantly judging the outcome of the outside world against your expectation for that world, and maybe forming salient memories when that expectation deviates greatly from the observation, and then sort of less of a memory stimulus if it's just moving along.
That's kind of our idea. It's like probably the most feasible mechanism is through some error correcting formulation. Right, you're observing some input, and because you've observed the input, not only does it propagate throughout your recurrent neural network, but you also have that input as like a reference to do some tuning with based on error correcting mechanisms. So we have explored more Hebbian types of learning algorithms to store these attractors and manipulations and whatnot, they're quite difficult to get to work robustly.
Awesome. Are there any other questions? We still have another minute. Jason, any parting words of wisdom?
Any parting words of wisdom? Yeah, basically this talk and everything that I want to say is sort of to promote recurrent neural networks. Sort of the computational power of having temporal memory. From a computational perspective, it's super cool because you get much more computing power and representational power. And what I'd really like to do is I'd like to shift our focus from thinking about neural networks and deep learning as a tool, right? Even if it's a biophysically motivated tool. And I want to start thinking about them more as systems, right? True neural systems and generalized artificial intelligence, et cetera. They have a lot of potential, and our brains are, as Jim said, they're just recurrent neural networks. We just don't know the architecture and form. So in my view, there's no reason why we can't understand, how each of the really complicated cognitive functions that we're able to do and manipulations we're able to maintain in our minds. There's no reason why we can't understand those in a neural context of mechanism and then engineer them. So yeah, all that to say, recurrent neural networks are cool. Memory is cool. Computational power is cool. It's not just input-output stuff.
Yeah, so basically to reinterpret what you're saying a little bit and to rephrase it, what you're saying is that perhaps in this course, we erred on the side of teaching deep learning as a function approximation sort of tool. Whereas the moment you think about memory, the moment you think about the temporal component, and the moment you think about this feedback and these paths, you really should be thinking of this as a system, a living, breathing, sort of interacting system, which has some state that gets constantly updated upon interaction with the world, rather than a function, approximator, basically. Right.
And it's the system that sort of, it's its own autonomous sovereign entity, right? And it's able to do a lot by virtue of having its own internal dynamics. And I think there's a lot of intuition, a lot of principles, a lot of powerful, both artificial and real applications in thinking about sort of, the system that is the neural network itself. That I think we have a lot of potential and could be very fruitful in the coming years.
But again, what you're describing here is an architecture that can be augmented at every one of those states with full CNNs, with full convolutional neural networks coming in from vision, from auditory, from all kinds of additional, interaction with the world, from the manipulation and actuation that happens with being an embodied intelligent system that sort of actually can not only make predictions about the world, but actually reach out and figure out how the world changes upon it. So we should be thinking of these cognitive systems, both in the, computer and in our brains, as these living, breathing things that have within them massive amount of function approximation power, but that are, and what makes them alive in a way is this actuation, memory, prediction, temporal component, etc., in the context of all of these subcomponents. But basically, in your view, at the core lies an RNN, and everything else is bells and whistles, basically.
In some sense, right? In some sense, I don't know how much more there is beyond recurrence that we have to think about to get all of the, human cognition. And that's so fascinating to me that you go from feed forward to recurrent, and it just seems like a small modification, but the addition of memory sort of absolutely fundamentally changes what a neural network can be thought of as. So that's what I would like to leave you with today.
Who are interested in sort of really having their mind blown about what's actually going on inside your brains, look at the hippocampus and the connections that it makes to so many different aspects of both our, central nervous system and the sort of peripheral nervous system, and sort of how memories are encoded through the co-firing of neurons across this living, breathing, interacting system. So I think, this is truly, truly very inspiring, Jason. I want to really thank you very much. And this actually concludes our journey through deep learning for life sciences. So I want to basically thank you all and sort of reflect a little bit about how far we've come. We've basically started with pure theory. Then we dove into genomics and genetics and variation and single cell and cancer and neuroscience and electronic health records and this incredible diversity of topics. And here we are back all the way to the beginning with all of the knowledge that we've acquired, looking at, using our own neural systems to look at how all of these cognitive systems that we have engineered allow us to start interpreting a lot of what goes on inside primate brains and maybe even human brains and our own cognition. So anyway, thank you, Jason, for wrapping this up so beautifully. And big thanks to Jim. And thank you to all for sticking through the class. Thank you for submitting your final projects last night. And we're so, so excited to see your videos and watch the recordings in our Thursday lecture. So big congrats to everyone. And thank you and see you on Thursday.